Hello everyone, in this lecture we are going to discuss how to explore the genome. The gene manipulation, we also call it genetic engineering or genetic modification. We all know right now, genetically modified foods or transgenic foods is common in supermarket or grocery. Genetically modified food, called GMO food, also known as genetically engineered food, or called bioengineered food, are foods produced from organisms that have had change introduced into their DNA using the methods of genetic engineering. So you could easily identify in this picture which one is genetically modified. And for GMO food tomatoes, it is bigger than the normal ones. For the salmon, if you raise them from 18 months, GMO salmon grow much bigger than wild type ones. Genetically modified maize is a genetically modified crop. Specifically, maize strains have been genetically engineered to express agriculturally desirable traits, including resistance to the pests and to the herbicide. The golden rice is a variety of rice produced through genetic engineering to biosynthesize the beta-carotenes, which is a precursor of vitamin A, in the edible parts of rice. It is intended to produce a food to be grown and consumed in areas with a shortage of dietary vitamin A. The vitamin A deficiency, which each year is estimated to kill 670,000 children under the age of 5 and cause an additional 500,000 cases of invisible childhood blindness. This gives us a hint that gene manipulation happens everywhere and every field. We will discuss gene manipulation in the next few slides. There are 8 GM crop species are mainly grown commercially, because several of them are major crops. Most current GM crops have been engineered for resistance to insects, the tolerance to herbicides, or both. The insect-resistant crops can turn genes from the soil Bacterium bacteriaceae. The protein produced in the plant by the Bt genes is toxic to a target group of the insects. For example, European corn boil or corn rootworm, but not to mammals. They currently grow GM crops in the US, traits for which they are modified, and percent of the total acreage of the crop that is planted to GM variety. IR is the insect resistant and HT is the herbicide tolerant. The DT is drought tolerant. VR is the virus resistant. So how to make a transgenic plant? First, you need to design and make the gene construct. Second, you need to introduce the transgene to plants by gene delivery technique. After that, you need to grow the transgenic plants. And also, you need to confirm the plants are transgenically by drug selection and gene sequencing. There are several essential components for successful plant gene manipulation. The tissue culture systems to grow them well, regeneration of whole plants from single cells, molecular gene construction technologies to make the new and right plasmid, molecular vector construct using foreign DNA, and also we need methods for DNA delivery. For DNA delivery in transgenic plants, mostly we use agrobacterial tumor fissions, microinjection, gene guns, sonication, efficient selection strategy, differentiation between cells which receive the transgenes from those that don't. Normally, we use antibiotic-resistant or herbicide-resistant genes as a selection marker. Only plants survive after the chemical drug selection will be kept. To do transgenic plants, molecular vector constructs were used to deliver foreign DNA. So in general, plasmid will have the promoter, which is used to control gene expression. Within the coding sequence is a candidate genes you are interested in, including herbicide-resistant genes, pest-resistant genes, salt tolerance, or nutritional enhancement genes like vitamins. Since the plasmid is the artificial gene construct, we need to add to the stop signal to stop transcription like the terminator. The plant transformation uses Agrobacterial tormificient. Agrobacterial tormificient is a widespread, naturally occurring soil bacterium that causes clone growth and has the ability to introduce new genetic material into the plant cells. The models for the process of agrobacterial mediate transformation. So this model depicts the various steps in the transformation process, including those occurring within the bacterium, perception of the phenolic and sugar signals from the wooded plant cells, various genes introduction, the T-strand processing from the Ti plasmid and subsequently exported of the processed T-strand and the virulence proteins, and those involving the plant cells or called bacterial attachment, T-strand or virulence protein transfer, cytoplasmic trafficking and the nuclear targeting of the T complex, and the DNA integration into the plant genome. So VIP1 is hypothesized to influence the event in the plant cytoplasm, whereas histone H2A1 influence event within the nucleus. In DNA macroinjection, also known as pronuclear macroinjection, a very fine glasses peptide is used to manually inject DNA from one organism into the another. For animal transgenics, DNA macroinjection is widely used. The most famous one is sheep Dolly. Dolly was a female domestic sheep, and she is the first mammal cloned from the adult somatic cells using the process of the nuclear transfer by DNA macroinjection. Beyond the agrobacterial-mediated transformation and DNA macroinjection, we could also use gene guns and sonications. It is not widely used in the field right now because of lack of efficiency. When you're doing transgenic plants, you could do overexpression, gene knockdown, or gene knockout. Overexpression usually refers to an experiment when DNA is added to the cells to force expression of the gene to much higher than normal level. Lockdown usually refers to an experiment that genes are deactivated or suppressed. The expression level of candidate genes is decreased. Gene knockout is the total removal or permanent deactivation of the gene through a genetic engineering. By DNA macroinjection, we could make transgenic mice, the transgenic chicken, transgenic cat. In those transgenic animals, people overexpress fluorescent genes. This is why they have green or red or yellow colors in the slides. The technique we use for gene knockdown is small interfering RNA called siRNA. Sometimes we call short interfering RNA or silencing RNA. It is a class of double-stranded RNA non-coding RNA molecules, 20 or 25 base pairs in length, and operating within the RNA interference pathway. It interferes with the expression of a specific genes with complementary nucleotide sequence by degrading mRNA after transcription, preventing translation, and thus decrease the gene expression level of the proteins. The lockdown algorithm or simply lockout are used to start gene function. So usually by investigating the effect of gene loss, researchers draw inference from the difference between the lockout algorithm and the wild-type ones. However, we need to distinguish between heterozygous and homozygous knockout. Heterozygous means only one of the two gene copies is knockout, while homozygous means both gene alleles are lockout. We could choose homologous recombination or site-specific nuclease to do gene knockout. For site-specific nuclease methods, right now in the field, they are zinc fingers, TALENs, or CRISPR enzyme cutting machinery. If we want to produce large amount of one proteins, we could do it in vitro by gene engineering. The protein production is the biotechnology process of generating a specific proteins. It is typically achieved by the manipulation of gene expression in the algorithm, such that it expresses large amount of recombinant genes. Commonly used protein production system includes those derived from bacteria, yeast, insects, or mammalian cells. So many proteins production includes three main steps, including gene cloning, gene expression, and protein purification. We use bacteria as an example to illustrate how it works. Here is the consensus protocol. First, we need to obtain the cDNA by amplifying full length of the genes through PCR method, using ligation to clone the full-length cDNA into an E. coli expression vector and get the positive clone through antibiotic selection in the culture dish. Express the protein in the E. coli called BL21 strain with introduction at low temperature in rich media. After that, solubilize and purify the proteins in a well-buffered solution, and then use affinity chromatography to purify the proteins. Get the high purity proteins. After that, we verification the purified proteins while G electrophoresis. Normally, we use SDS-PAGE as a method. SDS-PAGE is a variant of polyacrylate medium, gel electrophoresis, which is an elliptical method in biochemistry for the separation of charged molecules in mixture by their molecular mass in the electric field. It uses sodium aldosulfate molecules to help identify and isolate protein molecules. SDS-PAGE is a discontinuous electrophoresis system developed by Ulrich Lumley, which is commonly used as a method to separate protein with molecular mass between 5 and 250 kilodaltons. The procedure includes gel production, sample preparation, electrophoresis. After that, we do the gel staining and take images with results analysis. The SDS-PAGE can be either stained directly detected by antibody called Western blot. This lecture, we discuss gene manipulation technique. Here is some reference, and thank you for your attention.